we never seen this before, right? So we say, wow, this is big, right? This is important.
Like, come here, everyone, look.
Exactly. There's something fundamental going on, right? And this has not been appreciated before, right? So yes, that was a very important finding.
This is the James Cancer-Free World podcast. I'm Steve Wartenberg, and my guest is Dr. Zihai Li, the founding director of the Pelotonia Institute for Immuno-Oncology here at the James. Zihai is one of the world's leading experts on immunotherapy, which is a somewhat new and definitely a game-changing cancer drug treatment in which the body's T cells are energized to better recognize, attack, and kill cancer cells. However, immunotherapy isn't always effective, and one of the reasons is something called T cell exhaustion, which was discovered by Zihai and his team not too long ago. This is an important discovery, and Zihai will fill us in on this breakthrough and what it means going forward for scientists here at the James and around the world. Welcome, Zihai.
Well, thanks, Steve. Very, very kind.
It's good to have you back, and I'm very excited to learn about T-cell exhaustion. I think that name is perfect, and really you can almost understand it just from the name. But first, give us a little background on the very basics of immunotherapy and the role of the T-cell.
Right, I will answer that question, but before I do so, I need to clarify this. When you see my team, you mean everybody in the field, which is really the right way to frame it. And all of us in the field of immunology and cancer research, we're trying to figure out what the immune system can do or cannot do towards cancer. So very important collective effort from all of us moving this field.
That's true. Everyone knows what everyone's doing and builds upon it.
Exactly. It is the entire field. So the question's about T cells, right? So T cell, well, we know we have this defense mechanism called the immune system. So the first question is, what is the immune system, right? And I often tell my students, my first year medical student or graduate student, immune system is very simple. It's a whole bunch of molecules, a whole bunch of cells. But collectively, they do a great job in safeguarding us as a species to survive. And T cell is one of those components in the immune system. So T cell is a type of lymphocytes that is made in our body, in the bone marrow to be specifically. And then it goes to something called thymus. It's another organ that to be educated and to then develop. Then it go to, the rest of the body to patrol the system.
Patrol the system. I like that. It goes around and around.
Exactly.
Patrolling.
Yeah, patrolling. And if you find some bad guys like viruses, bacteria, mount attack, immunological attack. And guess what? This T cell is also critical for detecting so-called transformed cells or tumor cells. The tumor cells, they are different. They are cells that are transformed from our own tissue. So it is a different kind of entity, if you will, but that can be seen and recognized by T cells. Not only seen and recognized, the T cell can then attack those abnormal cells as cancer cells and therefore keep us cancer-free. So this is a very important component of the immune system to fight cancer.
But something happens with cancer cells so that T cells don't see them and attack them.
Well, very interesting question. Cancer development is not an overnight event, right? So cancer need to evolve from the very first event when the DNA within the cancer cell mutated, right? To then adopt this growth advantage to divide and to be able to spread. Throughout this process, they have to deal with the immune system. So as you can imagine, those so-called weak tumor cells, cancer cells, they are not able to evade the immune system. They'll be killed. So when we see patients with cancer, those kind of cells have effectively evaded the immune system. So therefore, looks like the immune system is not doing anything. That's absolutely incorrect, right? So the immune system has been doing things to keep this this person free of cancer for many, many years. It's when you actually have clinically detectable cancer, the cancer cells figure a way to evade the immune system. One way to evade the immune system is the concept you mentioned, that is T-cell exhaustion.
But wait, just to go back to something you
Yeah.
said, which is a new way of looking at it. When someone has cancer and it's diagnosed, those cells, the problems in their cells could have been going on for years, but the immune system and the T cells have recognized these weaker cancer cells for years and destroyed them. But then the
Exactly.
cells get stronger over years and time, and that's when their tumor grows and becomes a problem. We've never really talked about it that way, but that's a really clear way of looking at how it happens and why older people tend to
Yeah.
get cancer more than younger people.
That is very important concept. This is called, evolution, if you will, or immune editing, if you will. So this dynamic process. That's precisely the point we have reached collectively in the field, that during the entire process of, oncogenesis or cancer formation, immune system play a very important role in shaping, the cancer, what they are and what they can or cannot do.
But then we come across T cell exhaustion.
Right. So we said T cell a wonderful weapon that we got in our immune system and can detect and kill tumor cells. Great. Then why we again have cancer? It turns out when T cell recognize something and it can be activated, right? And this process is very interesting, from resting T cell to active T cells. But when T cell then finish the job, let's say destroy the virus, destroy some of the cancer cells, they then go back to the resting stage, so they can recuperate and be ready to attack next time the T cells see cancer again. But in cancer, which is almost like a chronic disease, people have cancer for a while, and the T cell being constantly kind of engaged, constant engagement without any break, without any rest. And I challenge you, Steve, if you run marathon continuously for 2 hours, you'll be okay, right? For 20 hours, that will be a problem for you. This is the concept of exhaustion. So T cells trying to run a race for a long time without any kind of rest, without recalibrate the system, and eventually it become sort of dysfunctional. And we use this exhaustion just for illustration. They are not as functional as the activated T cells.
So they still work, but just not as well,
Well-
or they don't work at all.
That's also a great question. They must be doing something. We're still trying to figure out whether so-called exhausted T cells still doing something, and some aspect of function can actually be seen. And then, if you can run some sort of assay in a laboratory, you can see they're actually doing something, but they are absolutely not efficient. Why we see that? Because one of the mechanisms, and and I think our viewers know, is related to so-called immune checkpoint molecule, PD-1, right? Now we know if we block PD-1, the so-called exhausted T cell can actually be rejuvenated. So in other words, they can actually be sort of recharged, reprogrammed, and become a functional cancer fighter cell again.
Can they also be recharged through the CAR T cell process? Or is that something separate?
Very, very interesting. CAR T, another concept is a chimeric antigen receptor. It's a synthetic receptor that can artificially give T cells specificity and function. But you're right. In order for those T cells to function, the CAR T cell function, they also have to play the same rules, right? Same biological rules, same playbooks. So yes, they can also be exhausted. If you constantly activate CAR T without break, they will also be exhausted.
So
That's
the
right.
CAR T cell process is when you remove the T cells from the body,
Right.
biologically change them and alter and make them stronger, put them back in.
Yeah.
They too eventually will
Yeah.
suffer from exhaustion.
Biochemically and genetically, you change them. But you're absolutely right. It still can be exhausted. And this T cell dysfunction or exhaustion is really a big problem that we got to overcome in order to have, effective immunotherapy.
So it just sounds like cancer cells are relentless and keep going and going,
Yeah,
and you can slow them down,
yeah,
but it's very hard to eliminate them because
yeah. This kind of fighting, and Tom and Jerry kind of fighting, is going on all the time, right? And once again, when patient is diagnosed with cancer, we kind of we got to conclude a little bit that somehow cancer win at that moment. So as a physician, as a cancer doctor, as immunologist, we want to help those patients, right? We want to give them some weapons, some sort of more kind of weapons, if you will, to help the T cell to be rejuvenated, so that we can till the battle, so that we can win against cancer.
Yeah, I don't want to, and I'm sure we both don't want to make it sound negatively that the cancer cells always win, because nowadays more and more you are putting people into long-term remission through immunotherapy.
Correct. Absolutely. No, this is really the goal. I think many of us, if not all of us, really believe that the war against cancer can be won. That is because we now understand how the game is played, right? So we have all the conventional weapons like chemotherapy, radiation, and surgery. But now we actually know that the self-defense mechanism, the immune system, actually hold the key. It's really to winning this war.
One of the things I've learned from listening to you and the other experts is the more you understand the biology of the body's immune system and cancer cells, the closer you are getting to that cancer-free world.
Yeah.
And this is one more example of understanding the biology of T cells.
Yeah. Well, this is why I'm so excited, and those of us who work in the field collectively feel like, we can do this. And absolutely, the more we learn, the more switches we learn, the more way we can actually flip some of the switches, so that the immune system can indeed be more powerful than is needed, actually, to get rid of cancer once and for all.
Okay. That was great. We're going to take a quick break, and when we come back, we're going to learn more about flipping the switch.
Thank you.
The best outcomes for treating cancer come from early detection and early diagnosis.
The James Cancer Diagnostic Center at Ohio State
is designed to provide rapid evaluation and a clear diagnosis.
We are transforming care by giving patients direct access
to cancer experts who study and treat cancer every day.
If you think you have cancer, the choice is clear.
Choose the James Cancer Diagnostic Center.
We're back with Dr. Zihai Li, and I'm just curious, what made you think of to look at the whole concept of T-cell exhaustion?
So there's been many groups, many labs studying this process. So the way we come in, we say, okay, well, it looks like many people have already studied all those, what I just mentioned, pathways. And there are actually not so many people who are studying what's going on with protein trafficking, protein synthesis, protein folding in exhausted T cells. So why that matters? As you know, eventually, I mentioned DNA, RNA, eventually you got to have a protein in order to function, to carry out some sort of function, right? So for example, if you want to move things, you need your muscle, right, which is protein, right? Your muscle cell, the fibers has to contract. This is protein. This is carry out the function, not DNA, not RNA. It's protein, right? For lymphocytes to kill tumor cells, you need also proteins, things called cytokines, things called, granzyme or whatever other molecules. They are all protein. And
that the lymphocytes have to have proteins in them, or they need to find the proteins in the cancer cells, or both?
Right. So the bottom line is that within the cell, what carries out the function is the protein, not the fatty acids, not nucleic acid, not DNA or RNA, but it's protein. So therefore, we really should look at the protein, right? And one thing before I even get to the heart of the question is that, we have a cell. Once again, we have 20,000 protein molecules in the cell, 20,000 different kind of proteins, right? It's a very, very crowded environment, right? So you got to find a way so that the protein do not bump to each other, and certain protein cannot be made too much. And so, in other words, you should have a very healthy ratio of the proteins, and the protein have to go to the right compartment. They cannot aggregate, adhere to each other, and that will cause problem, right? So fundamentally, it's actually something that life realized from the very get-go. When single cell organism emerged on the planet, the life realized we got to find a way to help protein folding. Protein have to fold it into a proper shape in order to carry function. So this is really, really very, very fundamental. And that aspect of biology was not really studied carefully before we begin our work.
So you focus on the proteins within the cells.
Exactly.
Okay.
Yeah.
What did you find?
So what we did was we took exhausted T cells, which you can isolate, you can make, and it also took so-called effector T cells, the cells that are actually functioning very well. So those two kind of cells, right, exhausted, not exhausted, and to see what are the differences in their protein composition.
That sounds so simple, but
yeah,
that's the breakthrough, to look at the difference.
Right. So we found all kinds of surprises. This story is full of surprises. So yes, indeed, there are differences in entire protein content of two different cells. And the question is, what are the differences, right? Just by sheer number of cells and what pathway seems to be increased, what path seems to be reduced, it looks like the protein involved in something called protein quality control, in protein folding, actually is increased significantly. Those proteins, by the way, oftentimes we call them stress proteins. They are involved in dealing with protein stress. Another term, just too many terms, they fall into this family of proteins called the heat shock proteins. So heat shock is a stress, right? Our physiological temperature is 37 degrees Celsius. If you have fever like 38, 39, you cause sort of you change the thermodynamics of the protein. You turn on this group of protein called heat shock protein. They are really very important in stabilize the structure of proteins. So it turns out in exhausted T cells, those proteins go way up. So that tells you something's going on with protein quality control, or maybe protein quality was not very good. That's why you induce this group of stress proteins.
So the number of stress proteins is going up, which makes sense because then the T cells become stressed and less effective.
Exactly.
And okay.
And to be even more kind of intriguing, it turns out certain stress proteins go up more than others. So we don't need to go through that much of details, but that tells you that this is not just every stress molecule go up. So there is some selective, specific family of molecule that has to do with T cell function that actually gone way up. So that's one observation that's very surprising. Second observation is that those proteins, despite the fact there are so many stress proteins that are involved in quality control, increase at high level, but the protein themselves, many of the protein substrates, if you will, they are still not folded in proper shape or configuration. So it turns out they aggregate, right? So they aggregate. Steve, let me ask you a question. How do you like your eggs?
Over easy.
Over easy, okay. Okay. I like to scramble my egg. So if you scramble your egg,
I like mine scrambled.
yeah,
If that's
exactly.
easy,
Your
okay.
egg scrambled. So what happens? You basically heat up the eggs, and then things aggregate. The liquid egg becomes solid, right?
Okay.
Solidify.
Yeah.
That's protein denaturation.
Oh.
That you destroy your protein, right?
They become,
They clumping, right? They aggregate. They become solid.
So-
yeah.
and then they can't function.
Exactly. That's exactly right. So that turns out it's happening in the exhaust T cells. It's like exhaust T cells being cooked. The clumps of proteins, aggregates.
So
Yeah.
take me back in time to when you're looking at you or someone in your lab, you're looking through the microscope
Yes.
and you realize this,
Yes.
that these cells,
Yes.
these stress cells are becoming solid. And I'm like, what was that like when you saw that?
Well,
I mean, that's big.
we never seen this before,
Yeah.
right? So we say, wow, this is big, right? This is important.
Like, come here, everyone, look.
Exactly. There's something fundamental
Yeah.
going on, right? And this has not been appreciated before, right? So yes, that was a very important finding. All right, that's so stress protein go up, but yet there are a lot of protein clumping, aggregation going on. So the system is collapsing.
So stress clumping.
Yeah,
Those are the
exactly.
first
Stress
two.
clumping. All right. Third thing, it's really surprising to us. Third thing, that is, when you have so much problem with dealing with proteins, right? There is a natural mechanism, natural defense mechanism that we know, natural feedback mechanism that's already been worked out. That is, don't make too much protein now. So when you have so many problems, right, naturally, you should slow down protein synthesis. Make sense? Right? Because your protein is aggregating. You have too many protein to handle. You cannot fold them into proper shape. Why make more protein? If you make more protein, you'll overwhelm the system. You make system worse.
Oh, it almost sounds like I would have said the opposite. You need to make more proteins because fewer are working. But you're saying if the body's already malfunctioning and making more proteins, they're just going to go bad quicker.
Yeah, exactly. But anyway, before the experiment, we thought that the exhaust T cell should slow down protein synthesis. Should slow down now because you cannot deal with so much proteins. It turns out your prediction was correct. It turns out they still continue to make more and more protein
because it realizes it needs to make more, but then they just turn bad.
The rate of protein synthesis,
wow,
actually goes up, way up, as opposed to down. So your prediction is correct, which in my sort of thinking prior to the experiment was a little bit counterintuitive. I thought the feedback mechanism is to inhibit, is to slow down as opposed to go up. But for T cells, for exhaust T cell, which is actually different from other cells, by the way, other cell will do exactly as I said. They will slow down protein synthesis, but in exhaust T cell, protein synthesis actually go up. All right. That has to do probably with the functioning of T cells, because after all, they still have unfinished business. Even though they're exhausted, they're still trying to fight. So therefore, they want to make more protein because some of the proteins become aggregated and not functional. Let's make more fresh protein. So by this kind of thinking, this is almost like a very determined T cells, very determined, want to really fight. So, but that unfortunately causes more problems, right? So sometimes, I just mentioned before, you can't run 20 hours straight. You probably should take some break, and then pick up and continue. Then you actually can run further distance.
Okay, so you discovered these three things about the cells.
Right.
So what does that mean? And let me take a quick guess, is that just you found it, that's not the solution. So you and all the other immunotherapists are now, for the next X amount of years, everyone's going to read about what you found.
Yes.
And now try to help the T cells overcome stress and exhaustion.
Yeah. Well, thanks for putting that way. Right. So I wanted the field to understand that the protein quality control is a problem. So yes, we can still study genetic mechanism, metabolic mechanism of T cell exhaustion, and so on. There are ways to probably tweak the system, make the system better. But you got to pay attention now to the protein quality control. You have to. If you do not solve this problem, I'm afraid all other strategies is not going to go very far.
All other immunotherapy strategies.
Exactly.
They'll
So whether it's
eventually-
CAR T or any other sort of immunotherapeutics we're doing, if you do not incorporate this into your thinking, into your experimentation, then you will not go very far. So yes, indeed, I think this is an important sort of chapter that people need to pay attention to. So then, more specifically, how do we leverage, right, this finding, and how do we then get a better, in terms of next generation immunotherapeutics? So I mentioned very briefly early on, the whole bunch of stress proteins, maybe even 10% of the protein are involved in protein quality control. And I said not every stress protein is the same. Some seems to be more important in driving exhaustion program. Some of those proteins are actually is important in safeguarding the cell so that they will not go exhaustion. So we found a whole bunch of molecules like that in this paper we published. So much so, I think we test more than 10 of those potential switches, if you will, right? And we found at least 2 or 3 of them. If we turn them off, or even just slow down their expression, that will make the entire system better. It'll make the, from this protein clumping helpless situation to actually more like functioning like normal cell, become all of a sudden useful T cell again. So I think this knowledge will help us to kind of manipulate some of the system so that we can improve the quality control, in a healthy way.
But how will you do that? Is that changing the formula of the immunotherapy or adding something to it that'll do that?
Right. So one way to do it is add something to kind of stabilize the protein quality control, right? There are actually some of the drugs out there that can help with this process. And genetically, again, you can manipulate some of the stress protein especially for the CAR T space, which is kind of genetic engineered cell. You can, instead of just doing the chimeric antigen receptor, you can add another switch there, right? So chimeric antigen receptor plus something else, right, to make the T cell more robust in dealing with this kind of protein stress. There are drugs which are fall into several categories. The drugs involved in inhibiting protein synthesis, drug that are involved in sort of get rid of the denatured protein. So those kind of drugs are being developed, and then I hope by combining some of this strategy with the existing immunotherapeutics, we will see improvement of our therapy.
Wow. So this is what I didn't realize when we started, that once you've identified these problems in these exhausted T cells, there are already certain drug treatments that can help. More are being developed.
Exactly.
And so you're all okay. So you're not starting from scratch and helping these stress cells. You already have some ways to do it, and you're developing more. Yeah. And as you've talked about before, with immunotherapy, the key is to find a target for the T cells, to help them find targets. Right. And you're discovering more and more targets all the time. So it's each new target you discover, this whole concept of T cell exhaustion will play into making that new immunotherapy better. Exactly.
I mean, that's a very nice way to put it. So, as a scientist, we're driven by curiosity, right? We're curious about nature. We want to figure out things. But in this particular context, when we study cancer and cancer immunology, whatever knowledge we learn by studying the behavior of those cells, the system, indeed it will come up with better strategy to manipulate the system so that we can actually help patients. So that is that actually makes me very excited, and it's very fulfilling career. You satisfy your own curiosity in an interesting way, but at the same time, whatever you find, whatever you discover, could be translated into helping patients.
Yeah. So let's look ahead 5, 10 or more years.
Yeah.
Even like when you were at the very start of immunotherapy, when people thought this was a ridiculous idea, but now we know it works. Now, when you combine immunotherapy with your new T-cell exhaustion and how to beat that, look ahead 5, 10, 15 years.
Yeah.
How much is this going to help?
Well,
I know
let's think
you're
about
a scientist and you're cautious, but
yes,
throw caution to the wind for a
That's
minute.
right, exactly. So the field of immunology is relatively young. So if you think about the first time the so-called immune checkpoint blockade was proved, it was 2011. So the field is like teenager right now, right? Teenager can do a lot of things, right? Sometimes not purposeful, right? So the field is a little bit unstable in that regard. However, this particular teenager, right, somehow is very intentional, very purposeful, and very smart with all those knowledges, with AI tools and everything. The field is rapidly accelerating. So this very, very teenager is very precocious. I think he or she will get a PhD in 2 years.
Okay.
Right? So you fast forward for 10 years, I am absolutely optimistic that a vast majority of cancer will be dealt with by cancer immunotherapy, one way or another. Right? Maybe not 100% of those patients, but a significant number of patients, very high percentage of patients will benefit from immunotherapy. I'm talking about every cancer.
Every kid.
Whether it's a leukemia, whether it's a melanoma, or even brain cancer for that matter. The reason is that this is a conceptually something you mentioned in your introduction. It's a new way of dealing with cancer. And this new way of dealing with cancer is the most important way.
Wow. Well, congratulations. This is big, and thank you for filling us in. And to me, what's really cool, you were brought here to lead the Pelotonia Institute, which you've been doing great. And this is just one of the many, many things that you and your big and growing team are doing. So what's it like to be at the forefront of and leading all these great scientists and connecting with the Pelotonia community who are helping you to raise the money to help you do this? This is like really a perfect storm.
Yeah, well, this is such a nice word, team, right? Even this very study that we just published, it's a collaborative effort. Well, lots of labs coming together and thinking together about this big problem, and figure out a way to advance our understanding of this process and hopefully come up with better strategy for cancer therapy. Absolutely. Pelotonia is such an amazing partner. I don't think I'm here, I don't think I could be here, to be honest, without Pelotonia. And I don't think we can do, as much as we have done without the support of the entire community, the entire university and Columbus, and so on. So I think this team building, team concept is not buzzword, is actually what's happening, right? So my laboratory is located in Pelotonia Research Center.
Yes.
Right, which is built by folks like you, right? With your effort, your contribution to the field. And again, I'm very excited. I'm not alone in fighting this battle. I'm together with everybody else. And that's the spirit that we have here in Columbus, and we can do big things as long as we work together as a team.
Well, every team needs a good leader, and your passion, enthusiasm is leading the way. So thank you once again, and good luck as you and others continue along the line of T-cell exhaustion and learn how to figure that out.
Thank you very much.
This podcast is brought to you by the Ohio State University Comprehensive Cancer Center, Arthur G. James Cancer Hospital, and Richard J. Solove Research Institute. For more information, check out our website, cancer.osu.edu.